Hello everyone and welcome back to Biology with Mrs. Evans. In this video we're going to look at the second part of basically protein synthesis. So protein synthesis starts off with transcription. Remember, transcription is the process that makes messenger RNA from DNA, and that happens in the nucleus. And then once messenger RNA arrives at the ribosome, basically the translation process is going to be starting. So translation begins again when messenger RNA arrives at the ribosome and when a start codon is read. So remember, there are three base sequences on messenger RNA called a codon, and those are what's going to correspond to the amino acids that are going to be used to make the protein. So, messenger RNA arrives at the ribosome. The ribosome is going to read it. Notice that this particular messenger RNA strand has this three base sequence. So, if it doesn't start with AUG, then the ribosome is not going to do anything with it. Once it reads AUG, it will start protein synthesis, and it will continue protein synthesis until it reads a stop codon, and the stop codon basically tells the ribosome that the protein has been made, and then to release the protein that was made. Transfer RNA, once it delivers the amino acid, as you see this one did, it's delivered. Here it is. It's binding to the next one, and then this one can now go and pick up another amino acid that corresponds to this sequence. And then messenger RNA, once the protein is made, then the cell is going to break down messenger RNA because the protein has been made. So let's look at how translation works. So again, once a start codon is read, the ribosome basically is going to read it, and transfer RNA is going to bring over the corresponding amino acids. And again, it's reading it in three base sequences. So you can see here, this is the polypeptide chain that is being created based on this messenger RNA. So this particular codon codes for this specific amino acid, and it will continue until it reaches a stop codon. Once a stop codon is read, then basically the protein will be released. This is going to be, messenger RNA will be basically broken down because it's no longer used, and then transfer RNA is free basically to go and pick up amino acids that correspond to the anticodon that is located basically floating around in the cytoplasm. So in order to translate messenger RNA, you guys are going to basically play the role of transfer RNA. And in order to do that, you need to be able to use a codon chart. A codon chart tells you basically the amino acid that a specific codon codes for, and there are two types, as you see here pictured on the screen. I have no idea which one you're going to get on the SOL, so you need to know how to use both of them. So I'm going to teach you how to do that here in a second. Alright, so this is one type of codon chart, and the way that you would do that is you start basically with your first nitrogenous base here. So let's say, I'm going to write it here on the screen, let's say we want to know what UCG codes for. Alright, so the way that you do this is you find your first nitrogenous base, which is here, so that's U, and then you look for your second base up here, which for us is going to be C, and basically you are going to come where those two meet, which would be here, so U and C meet in this box, and then you're looking for your third base over here on the right, so we're going to go down to G. That's our third base, so this would this codon would code for serine. Alright, so we're going to say that that codes for serine. Alright, so again, the process is you find your first base and you use this column here to find it. And then the second step would be to find your second base here, come to where the two match together, alright, and then find the third codon here on the right and basically read over. So let's try another one. Let's say we're coding for CAG this time. So CAG. Alright, so again, first thing, here's my first base. All right, here's my second base. So I'm going to come to where the two meet, which would be here. And then I'm going to find my third base, which is the G, which is right here. So this one is going to code for, I think this is glutamic acid. All right, so that would be what CAG codes for. So again, remember AUG is a start codon. It codes for methionine. It tells the, it tells the ribosome to start protein synthesis. And then notice that there are 3 stop codons. You don't have to memorize those because they're always going to be on the codon chart. So protein synthesis would continue until it reaches that stop codon. Once it reaches that stop codon, the protein has been made, it's going to be released. Messenger RNA will basically be broken down because the protein has been made, it's no longer needed, and the process would continue when another protein needs to be made. All right, so this is the other type of codon chart. I particularly find this one to be easier than the other one, and a lot of you probably will too. The way that this one is you start in the center. All right, so let's say for this one we're going to code for UGC. All right, so for this one, you simply start in the center and you work your way out. So UGC would code for cysteine. Okay, so let's try another one. Let's try CCA. So again, you start in the center and you work your way out. CCA would code for proline or proline. So this one, I think it's easier. So you start in the center and you work your way out in that particular quadrant. So here is G. Let's see what GCA codes for. So we go GCA is going to code for alanine. So again, you're going to be responsible for looking at the different types of codon charts and figuring out what a specific codon codes for. We will practice this in class because it's a little bit hard in case you have questions. So again, codon chart tells you the different types of basically amino acids that are created by the codons. So if you look at the codon chart, you'll see that there are 20 amino acids that we know of. So there are 20 amino acids that are out there, and basically there are 64 codon combinations as to how those 20 are going to be coded for. You'll see that several of these have more than one codon that would code for them. For example, you see alanine has 4 here, serine has 4, and then you'll see, let's see, threonine has 4. There's a couple of them that will have 6 because they're in different places on the wheel. So, example, argine has 4 here, but then there's another argine here that has 2 more codons that will code for it. So, again, 20 amino acids that occur in nature, and then there are basically 64 different possibilities to how those 20 can be coded for. So the big idea, again, about protein synthesis is that DNA is used to make messenger RNA, and messenger RNA is used to make a protein. A lot of the analogies you'll see is they compare DNA to a cookbook. Messenger RNA is to the recipe in that cookbook, and then the protein that is being made is basically the food that you create. And my analogy is basically you go into a restaurant, you know what you want to eat, but the restaurant is not going to allow you to go back and cook your food. So basically you are going to be basically the DNA, and then you are going to relay the message to the waitress as to exactly what you want. She's going to copy that down, she's going to transcribe that basically, and she's going to send that message into the kitchen. The kitchen is going to be basically the ribosome. The ribosome is going to make that protein and is going to make your food. So that's my analogy as to how the process works, because when you go to a restaurant you can't go back there and cook, but whatever message you send back, hopefully if they copied it down correctly, you're going to get out in a short period of time. And that's exactly how basically protein synthesis works. So transcription, it starts with transcription. The information is copied from DNA and it's sent to the ribosome via messenger RNA, and then the ribosome is going to read it. Transfer RNA is going to bring over corresponding amino acids until basically a stop codon is read, and then the protein is going to be released. Messenger RNA will be broken down because it's no longer needed because the protein has been made. So here is another visual between, or the process of how a protein is made. So we have transcription and translation. So you've seen a couple of these that I gave you in a handout, but let's go over this particular one. So A is pointing out basically the nucleus, and we know that messenger RNA leaves the nucleus, so this is going to be messenger RNA. It arrives at the ribosome, so that's what C is. Here's our ribosome, okay? The ribosome is going to read it, the messenger RNA. So on this particular one, this is going to be G, all right? This is messenger RNA. And remember, a 3-base sequence on messenger RNA is called a codon. So there's one codon, here's another codon, UUC, and here's another codon, AAA. And then you have the transfer RNA, which is D. And remember, I remember it as a trumpet. And this one does really look like a trumpet, as you can see. This would be the mouthpiece. This would be where your fingers would play. And then this would be where the sound would come out. So located on transfer RNA, remember, is the anticodon. Remember, anticodon is a 3-base sequence that is found on transfer RNA. And it's going to match up to the codon. So you can see this one is going to be the next one to come in because it matches up with the AA. And you would look at your codon chart to see what AA codes for. We'll try to do that in class. So, and then this guy right here, this is our amino acids. So remember, there's going to be a bond that forms between the amino acids called a peptide bond. And basically, each amino acid will be added and the bond would form. Once the bond has formed, then the transfer RNA is free to go and leave the cell, leave the ribosome and go look for other basically amino acids that would correspond to that particular anticodon. So that is basically translation in a nutshell. Remember, it's 2 processes. It's transcription, or how proteins are made, is two steps. Transcription, messenger RNA is being created from DNA in the nucleus. It travels to the ribosome, where the basically the ribosome is going to translate it. It's going to use that information and make a protein. And again, we'll go over this in class, and you'll do a lab and see how this process works. Thanks. Have a great day. Bye.